All right, so we're going to talk about metagenomic binning, which is another service in BV-BRC. For the purpose of what we do with the binning in BV-BRC, it's been targeted at environmental samples, in large part because that's where the folks who are doing this, where their interests lay. And the intent initially for this, the service, was to use environmental samples to extend the set of genomes that we know about by extracting high quality, fairly complete genomes from samples. So the reads that can do this have a couple of characteristics. They need to be deep enough that they've got coverage that we can find the full genomes within them, but small enough to still assemble them. There are soil metagenome read sets that are terabytes in size that we just don't have a hope of assembling without a whole lot of work. Given one of these samples, this is the pipeline that we're going to go through. We'll start out by assembling the reads. And for this, we use one of the standard assemblers. We use Metaspades or MegaHit. We partition the results into bins, where each bin is going to describe the distinct genome. We do an analysis of these bins so that we keep just the ones that have genomes that are fairly complete, not too contaminated, and thus fairly accurately represent what might be a new genome. Like Andrew was talking about assembly, then these are all hypotheses too, that we have reason to believe that this set of contigs represents a genome. And so as a part of this, it's important that we can determine whether or not a genome is good or not. And so the analysis of the quality of the genome is important in this work. And we'll construct a set of bad genomes, and we'll give those to you, but we distinguish the good from the bad. So the measures of quality for the bins are two main ones. One is whether or not they're consistent. Do the set of proteins that we see in this annotated genome reflect the functional roles that we believe are there? Do they make sense in context? We also want to take a look at completeness and contamination. That if we look at genomes in a part of the tree, we'll find that there are sets of roles that if you have a certain set of functional roles in the genome. Again, I said, I'm speaking in shorthand. I should take a step back and say that I'm a computer scientist by nature, not a biologist. So all the biology I've learned is by osmosis. When I say a role is in a genome, what I mean is that the genome has proteins that we've annotated to have that functional role. I think it's genomes as sets of roles, right? I have a genome that can perform a set of functions. There are sets of roles that will always occur together in parts of the tree. And so we look to see if our genome has two of these roles and we always see three roles together and only have two, that's an indicator that the genome may not be complete. So we've got a set of tools. One is called eval-con, which measures consistency. One called eval-g, which measures completeness and contamination. And we'll see reports of those when we go through. And these are both part of the annotation service. Why even do binning at all? I was asked to speak quickly, so we do this because there's useful data out in the sets of reads in the environmental samples, and we like to glean what we can for genomes that we can't otherwise culture. Why use our service for it? This slide bothers me because it starts with the disadvantages, but you can only find genomes that are close to what we already have because we bin the genomes based on starting on contigs we already have in the system. We can find fewer bins than some of the competing tools. We can read about that more later. Although the advantages of, this is very convenient. You just upload your data and your bins come out. And just like anything else that we annotate in the service, the bins are annotated and integrated in the database. And you can do all of the other analyses that we do in BV-BRC with them. We've run many thousands of samples through. Let's see, as of when these slides were put together, we've done 23 gigabyte reads that we've assembled and binned. Came up with 22 good bins and 36 others. I don't recall. This is probably a gut metagenome, I think, is what they were running there. How it works. So we first look through the contigs for any sequence that contains a given marker gene. Here we're using PS gene. That was chosen because it tends to occur once in nearly all genomes. And so it's a good marker for the contig containing that PS, probably a representative of a particular genome. And we can identify the genome based on the closest PS in our database. So we use the reference genome for that marker gene. And we've got some parameters on what we call high quality references that we use to generate these. We have a binning process that finds the sequences from each contig and put them in the closest bin, if any. Then we annotate and check for quality. Suspicious contigs are removed. Then any bins that we have left over, we search for virus DNA. We use check V for this from the folks at IMG. And we annotate those if we can. There's a fairly small set of viral families that we can do annotation on. We haven't talked much about viruses. I don't think we have viruses on the agenda for the workshop, but folks are interested, we can chat a little more about that offline. The trouble with viral annotation is we don't have a single comprehensive annotation service for it. There's some of the families have better support than others. And some of the viruses that will come out came from a big project from the IMG folks that gathered a huge number of viral contigs from metagenomes and gave them names like DTR 1046. We don't tell you anything, but it just tells you that we've seen it before. All right, so I'm going to flip to actually doing some of this. So this will all look very familiar to you because we're going to start in the tools and services over here in metagenomic binning.
One concept that I think is worth mentioning, a lot of people will see nowadays MAGs.
Metagenomic assembled genome?
Yeah, and that's what the service is giving you, right? It's giving
Right.
you metagenomic, it's just called something different, right? So you'll see MAGs all over the place nowadays. People,
Well, binning is the act of generating mag.
right. People will recover genomes from metagenomic samples and publish, that spectrum of organisms and the assembled genomes that are from wherever, from the swamp or
Right.
from or whatever.
And there are many, many of them in BV-BRC now because they're in GenBank and we pull GenBank.
This service actually predates even the term MAG, right?
Right. By now, this interface probably looks familiar to you. The binning service will take either reads or assemble contigs. Remember what Rebecca said earlier about that sometimes you may want to do assembly outside of one of these combined services. So if you did your metagenomic assembly using the assembly service instead of this service, you would be able to select assembled contigs and bring the input in that way. We'll start with starting from read files. So the example here, we're going to start with an SRA ID. We're going to use the Ricea example. Got to go back to my cheat sheet here, which has an SRR ID in it. Oh, this is the TC flies. That's right. So this is a sample that's what the TC fly with its endosymbiont, right, Rebecca?
Mm-hmm.
All righty. So we're going to start out, find my window, just like with the CGA service. I put the run accession in the run accession on slot. I clicked over there to add the library. And so there's a few parameters that we can choose here. We can choose an assembly strategy, which is either Metaspades, MegaHit, or Auto. To use Metaspades, we'll see higher quality assemblies usually, but there's some tight restrictions on the data you can feed it. At least as the version we're running against now, you can only give it a single paired end library. If you've got single end library, or if you've got multiple paired end libraries, you can't use Metaspades and you need to use MegaHit instead. It's easiest to choose Auto because the service will then detect if you've got input that's suitable to send to Metaspades, it'll send it to Metaspades. You can choose under organism of interest whether you're interested in just bacteria, just viruses, or it'll try and do both. If you do both, it will pull out the bacterial bins and then feed what remains to the viral binner. If you don't bin bacteria, it will take all of the reads and feed them to the viral binner. So if you've got data that may be mixed and you're interested in the viruses, I'd recommend running it twice, once each way, because you'll get a little bit different data if you do that, a little bit different results. Pick an output folder, output name, and a genome group name. We haven't really talked a lot about genome groups. They define sets of genomes that you can use later in the analysis. If you give it a genome group name, it'll generate a group that will be the set of bins that were generated. I'm not going to go ahead and submit this because there's quite enough load in the system right now with all the jobs that have been running. So I will do a Julia Child thing. I also wanted to point out that there's another job that we would want to run. Remember when Rebecca talked about aligning the reads, that she took this mixed sample and aligned to a reference. One of the things that we can do is to take this data that came out of the aligner workshop folder. So we've got under our reads here, I'm back in the metagenomic binning area of the workshop folder. These are the read files that came out that were aligned to the reference. So just like with these other tools, you can pick read files and it's not going to remember where I was. So we pick both of our read libraries there. We click the arrow to send them over. I'll get rid of my SRR reads. And actually, this is something that I want to comment on a little bit, that in all these boxes where we've taken input data and clicked on the arrow button to put them in a list here, there's a little icon here that if you click, it will tell you in more detail what it is that you shoved into that box, because it gets truncated to display in the box. So you can double check that you've put into the box the thing you've expected to put into the box. And like I did with the other read set that I didn't want to have there, if you click on the X, it will get rid of it out of that box. So I'm going to pretend that I've submitted all of these, and we're going to go look at some results. Into our workshop, binning. The other thing that we would have done is taken our full data set and run it both against Metaspades and megahits. So I'm going to go look at the Metaspades results first. So this is our result from that SRR ID, the Metaspades unmapped run. Okay, so this is the entire data set run through Metaspades. And so this is what the output of a binning run looks like. You can see again at the top here, we've got a report that I will go through in a moment. And you'll see a bunch of files that called bin1, bin2. Here we've generated 2 bins, and these are the data resulting from those. One's an FA file. This is the contigs of that bin. So this is the genome that was extracted for bin 1. This guy, because it has a checkered flag, is the job result for that bin. And there's also some statistical information about the bins. Contigs at FASTA, this is the result of the assembly. So this is the assembled data that came from this run. So let's go look at the binning report. So this will give us a recap of the data that we ran. So this is the unmapped reads. And here we get the summary of the bins. So what this is showing is that we found 1 good bin and 1 bad bin. And for each one, we get a score that I don't recall the metrics for it. We'd have to look up in the reference to see how they score it. We get the genome ID that was assigned for this bin. We get the genome name. The genome name is derived from the name of the reference with clonal population added at the end. We get a reference to the reference genome that was used to form this bin. And then our statistics. So coarse consistency and fine consistency are a little bit tricky. So remember when I was talking about the fact that for each taxonomic group, the roles in the genome tend to cluster together. Say we're looking at glycolysis, right? The genes in glycolysis in part of the tree will always, say we always have 10 genes that make up that part of glycolysis. We build a machine learning classifier where we basically remove one of those genes and train up a classifier to say how well we can predict the presence or absence of that one gene based on the presence or absence of the other 9. And we run that classifier against the genes in our bin, and we get a score that says, the coarse consistency says that, how many of these did we predict presence or absence correctly? Fine consistency says how often did we predict the right number of genes, because we may have genes that are multiple copy. So, this genome did quite nicely. 95% of the time we predict the presence or absence. 94.8, we predicted the correct copy number. Completeness and contamination, we have essentially an optimized version of the CheckM algorithm that uses the data from the annotation. Here, for the Wigglesworthia, we got basically a complete genome with no contamination. We got 13% hypotheticals. This is the same as the count back in the summary counts from your genome. With the other things, we pull out the number of hypotheticals. We have 4 contigs, and with the rest of our statistics here. I'll talk about the stuff at the end in a moment. Our bad bin was a rickettsia. Remember this? So this is interesting because this is part of the symbiont. We had good consistency. We had good completeness, but it was contaminated. Over here, this other yellow flag shows that we did not get a good FES, so that we didn't get a good match to the reference.
It seems kind of odd that you didn't get a good FES if there was 99% completeness. Is that, maybe I'm missing something there, but that's.
I think this may go to the quality of the FES in the chosen reference. Because we go back here.
I also recall that I did from mapping against that rickettsia felis genome, and the line reads to it, and then, as I recall, it came up. So
Bob, maybe when we are showing blast or something, one of the exercises we can do.
Oh, pull the
Take the
PS
PS
from,
that particular gene and see.
Yep.
Actually, it might be there in the genome quality report also.
yeah, when I go click through to that, we'll take a look and see. So remember, this is the run against Metaspades, right? So let's go look and see what the corresponding mega hit run looked like. So this is the same input. So we see we get a different result here, right? We still got our two bins. We've got, I think these are the same consistency numbers we had before, but we had a whole lot higher hypothetical percentage, things that make you say,
And this time it was able to get,
yeah, we got a good FIAS down here. So
I generally try everything. I want the best thing I can get. So every possible combination I do, which you'll get to see tomorrow for variation.
on the topic of trying everything, remember the other thing we did in this example was to map it against a reference. And so we're going to look here. This result is the result of running binning against the mapped reads out of the FastQC service. And here, sort of as expected, we only have one bin. We don't have that other bin here because we pulled those reads out by mapping to this reference. We again have, just about the same consistency numbers as before, a little bit different, which is interesting, right?
It's mega hit this time.
Oh, this was mega hit.
Yeah, I don't know.
It's in the title. Okay.
There's my point. Name it.
Yeah.
So you know what you did.
Nobody's shown this yet. So I'm going to go looking for a job. So if you're looking for a particular job under this all services, you can click this and it will give you the jobs that correspond just to a given type. Here we go. Let's look at this one. I'm going to click on the job. I'm going to click view. So we've got our job results, but what I'm going to do is up top here, if I can make it scroll, there's this drop down here that says parameters. This is sort of the, looking under the hood to what's happening when what's talking to the back end service. But if you are interested, this sort of is the complete description of this job. It's an encoding of all of the parameters that you filled in when you submitted the job. So here we can say, there's also a bunch of things here that have defaults that you didn't see that the interface filled in for you. But we can see that, here's the output file was unmapped Boselsworthia. And here we see assembler is mega hit. So this sort of verifies that this really indeed was the mega hit run because that's what we told the back end to compute on. And we'll just go back here. So we're going to go back and look at the Metaspades output. And we're going to look down at the binning report. Over on the right here is something called potentially problematic roles. So this is delving a little bit deeper into why we have less than 100% consistency on the genome. So if you click on this, this will take us to another report. If you were paying close attention before on the things that we didn't see Jim click on in the genome output was a file called genome report.html. And what this is like the other reports, this is a detailed report on the output of the annotation of the genome. This is reported for every bacterial genome. We don't generate one of these for phages, I believe, is what we discovered. But this has our genome name. Again, recapitulation of the columns from another report, completeness contamination. We get what's called an evaluation group that is a signifier for the set of genomes that this guy is being compared to. And I will keep talking about the functional roles. This is showing that the classifier predicted that there should have been 1,300 and some roles for this guy. There were 34 roles that we had too many proteins for. There are 36 roles that we didn't have enough proteins for. And we also have the number of roles that we use for predicting completeness. And the meat of this is a list of, on a per functional role basis, the roles that had problems in this organism. So the two main columns are predicted count and annotated count. The predicted count is what the machine learning model says that this genome should have had. The model said that in the comparison group that this genome mapped to, we should have had one of the 16S rRNA, and we didn't have any. And over here in the comment, it tells it that, well, in the reference, we had this particular protein here does perform this role. There's a protein in our genome that's the closest one to it, but for whatever reason wasn't annotated. And so now if we want to, we can go click on this guy, take a look, and it was annotated as something else, right? And so This really doesn't tell us a lot yet, but it lets you, say you are an expert in, the 16S what's it. This may give you a handle on, starting to look at why this genome is only 95% complete, not 100% complete. We have a number of them here that were not predicted to be in the genome, but they were found. One interesting thing here is we've got the set of them that were not predicted to be here, but they're found and all found in the same contig. Things that make you say, well, maybe this contig shouldn't have been in this genome. Let's find some more interesting things here. Here's one that there was supposed to be one, but we had two of them. So let's just go click on this guy. I'm going to click on the two features. Widen this up a little bit. When you get a genome out of the annotation system, it numbers this last digit in the feature identifier is sort of the index of that feature along the genome. And we see that these two are right next to each other. And if I go look at the compare region viewer, what we see is this display that shows here's this GTP binding protein here. And this protein here are the same function, but they're broken up, right? So for whatever reason, this protein was split. And so perhaps there's an assembly error in here, perhaps frame shift, who knows? But this is a hint that there's potentially an issue with the annotation of this gene. I think we've covered most of what we were going to cover in here. Oh, the other thing related, remember I was talking about how maybe we had a wrong contig in here. There's a summary down at the bottom of potentially problematic roles by contig. And it turns out one of these contigs had 37 features that were problematic, right? And there's one other contig that had 2 that were problematic. Although this one is 550 kilobases long, so.
And after
yeah.
I saw that, although this is an endosymbion.
Well, how big is this
When I
guy?
saw that, I would be thinking, ooh, maybe lateral transfer.
Yeah, but that's also the biggest, that's probably the biggest contig, so
Well,
yeah,
that's why
so.
I would be
yeah.
thinking
Because what-
it's an island in there.
This is actually a pretty nice genome as far as the quality of it. What you'll see in some of the genomes that are worse assemblies is that you'll get entire contigs that are clearly not part of this genome that got binned in for whatever reason. That will show up as that all the genes on this contig don't belong. And so if you're doing a nice tight annotation, you probably delete that contig
Just
from the
pull it
genome.
out.
Just pull it out.
Wigglesworthia's an endosymbion.
Yeah, but is it likely or is it not likely?
They're supposed to be not have a lot of interactions to mix well and play well with others because they're embedded
Oh, they're
within
embedded.
the host cell.